Good morning, everyone. Fourth lecture, Biol3157. Today we're going to talk about trait loci, which is a phenotypic measure that's heritable and can be detected by SNPs, specific SNPs. So first let me introduce a little bit. What we're doing now is forward genetics. It's actually a combination forward and reverse genetics because we have all the potential SNPs ahead of us as candidates, and we're going to go now testing each one of these genetic variants for a direct association with a phenotype. So Mendelian phenotypes are these large effect, major gene traits. We can often see them segregating in 1 to 2 to 1 or 1 to 3 ratios in a parent offspring. Usually we can identify the causal variant, certainly SNPs that are very close. And then the reverse genetics, we're using genome sequences. We've got lots of anonymous SNPs. Some of them may actually cause a stop codon or frame shifts. And so we might have a prior on which SNPs may be more informative. However, there are many correlations among SNPs as they're inherited together in a block. So sometimes you can't get at the causative SNP. And this type of study is called genome-wide associations. We're going to take genotypes that we have and phenotypes that we measure and look for a direct link which suggests that these are heritable traits. So there's several things now that are describing a very similar approach. So early on, before we had whole genomes worth of data, we often had special mapping populations and hundreds of markers that were each analyzed individually on gels in large labs. And that was the beginning of quantitative trait loci mapping, QTL mapping. Probably started in the late 90s, but really was popular in the first decade. Then genome-wide associations kind of surpassed that. This is high-resolution quantitative trait looks mapping, and we went from hundreds of markers to millions of markers in quite a short time. The limiting thing then became the availability of recombinant samples and getting diversity. So that's what we talk about population restructuring, and that's how we began the class talking about samples. So now we phenotype those samples and identify the causative variants, and that allows us to make predictions about what other samples will look like based on the relatedness of those samples to the test data. It's a very exciting ability now to predict the phenotype from the genotype. You can use a seed to know what it'll look like, or even a child, what they'll grow up to look like for certain heritable traits. Now, if you want to use that predicted information to determine which seeds to go in the ground, that's called genomic selection, and which seeds to breed with each other. So these terms you should be familiar with. Now, they all require and assume that some fraction of the phenotype variation is heritable in the next generation. And if this is only 10%, then your predictive power is not going to be any higher than 10% at the most, and your ability to select may only be 10%. But if that gives you an edge and advances the breeding population, it's certainly worth it. In large studies, if you can make a 10% better guess, those actually are quite high heritability traits these days. And so missing heritability and invisible heritability are what we're trying to capture with these markers and specific studies. I'll talk about that more in a minute. So just the background, we've been doing genetics since the 1860s. Mendel and Darwin came up with this genes and selection ideas. It really got explicit in the mathematicians in the 1920s, Haldane, Fisher, and Wright. And they had this idea that there's many genes of small effect that would each control traits, but they would be passed on in blocks and in a mixture model into the next generation, that the effect of many genes of small effect would add up to high heritability traits. Then we got the structure of DNA, and we could understand about recombination and how they would be inherited. And that opened up kind of the era of molecular biology, where we were trying to track down the individual causative mutations for genes. This was the beginning of gene cloning, a very exciting time. We always had phenotypes before we had genotypes, and now that's beginning to change. We have um, many mutations, then we could have genome sequences and make collections with single gene mutations. This happened about the same time as we began to do serious quantitative trait locus mapping and line crosses. And this was a way to try to clone natural variants by using um, mutant collections as candidate genes, where the genetic architecture is just much simpler. In wild populations, there's so many changes, it's hard to identify the causative ones. And so by the 2010s, we moved to this era of genome-wide association, where we're both identifying these major genes that segregate often in Mendelian ways, and also the contribution of minor genes. So it's taken a century or more for theory to meet practice, and now it's a routine approach. So what we've learned in the last 5 or more years about heritable traits, this is after scanning lots and lots of populations at lots and lots of markers and lots of phenotypes measured, that some highly heritable traits are in that 30 to 40% of the phenotypic variation can be explained by what you're born with, the nature, the genes. And the rest is just noise. That's nurture, that's environment. And even traits down at 10% would be considered heritable, sort of low heritability, but still able to be dissected with these approaches, especially in large studies. And human population studies are now often in the tens of thousands of samples, and those are the numbers that we're shooting for in plants. Certainly the field breeders look at tens of thousands of plots, and they do meta studies and many plots aggregated across sites and seasons. And what we're finding then is, after you scan for SNPs, that there's usually a set of large effect. And now these terms, small, medium, and large, are really relative to the economic feasibility of the study. And the modern studies now are in this tens of thousands of samples and millions of markers, and the phenotype variation to go with it. So when these data sets are assembled, you can scan through, and we find a few kind of outlier genes with large effects. 1 to 10 genes that could explain 1 to 10% of the variation. And then medium effect genes. There's usually tens to hundreds of these medium effect genes, which, at most explain 1%. So it's not really going to change what you've inherited at any of these hundreds of genes. They give you a marginal predisposition, if you will. And then with high power studies, we can even fit another 10 to 1,000, 100 to 1,000 genes with really small effects. But the data sets are there to estimate them and include them in the models. You're not going to be very confident on exactly which 100 to 1,000 genes they are, but they're likely to be these small effect genes that are not noise. There's even another category of very small effect genes that have been identified in very large studies. And you wouldn't, really diagnose somebody as being susceptible or not based on a variant in any of these small genes. But when you add them all up together, this is the power now, is to have markers that do give you a suggestion as to your predisposition. And when you add them all together, you get tens of thousands of SNPs out of millions that are being tested. So this is still only 1% of all the potential variants that would have any minor effect at all on the phenotype. But when you add those together, we can get that 30 to 40% phenotypic variation explained. And so this has largely resolved the earlier question of missing heritability, where we know that human height, for example, segregated within families, the parent-offspring regression is high, 60 to 70%. We know is inherited However, we couldn't identify the precise genetic architecture because of these many genes of small effects. And it wasn't until very recently where we got studies in humans anyway, in the hundreds of thousands of samples, that we could start to predict how tall you'll be. And that's for height, which is rather high heritability, but many genes of small effect. Now, there's always these large mutations that segregate in specific families that can be even more important, but they're basically never exist anywhere outside of those specific families. Now, in a plant breeding context, identifying these very rare but very large effect mutations can be very tractable, and then you take those variants and you sweep them into breeding populations. So, I'll talk about invisible heritability a bit later, but that's something that's never been properly measured before. And as we're getting better and better with this parallel discipline of phenomics, we can start to squeeze out more phenotypes and suites of phenotypes that could also have a genetic basis that haven't really been looked at yet. All right, so this data sets always begin with the samples by SNP data, but now we're going to merge in samples by traits, and you'll do this in the exercise. The traits are candidate traits, really. They're potential phenotypes that you measure that have potential heritability. And if you find a few SNPs, then you could call them traits and be able to make predictions about what the next generation will look like. Now, these studies are very powerful if you have a well-controlled design, and we measure the phenotypes in a common garden. And so plants are really great here because you can put all the seeds together in a common garden. It's much harder to do for animals, but you can imagine cages, certainly for mice, or tanks for fish, and corals, and all kind of other wonderful things that you can measure the phenotypic variation in standardized environments. And that's the best way to get that to genetic basis. Now, previously we talked about landscape genomics, where we're looking for allele frequency differences between sites or that correlate with environmental variables, and you've got a lot more noise and a lot more structure in those studies. Now, that's Mother Nature's studies. They're big and old and almost random, but when we design a powerful experimental study, especially in plants when they can be well behaved, and it's not like just a survey of, say, how often you get anxious, that these behavioral phenotype surveys that are now common in human studies are really full of noise. But that's what exists in the world. Most of the variation out there is not genetic, it's environmental. And then you can do something about that. These lifestyle, it's not nature that you're born with, it's the nurture in your lifestyle that control much of the phenotypic variation. But nevertheless, there could be small predispositions. And in these well-designed studies, we can go after it. And usually these studies are replicated in multiple blocks. You can put the same seeds in different common gardens, and you might apply different treatments and also measure individual plants and individual lineages multiple times, and for multiple phenotypes that may be correlated. So these are the powerful experimental designs. You start, first of all, Mendelian traits, cross a red plant with a white one, and you might get pink in the next generation. You can back cross it, or self-pollinate and see the segregation of these major genes. Now, quantitative traits are more environmental noise, multi-genetic effects, and so it's not simple discrete classes of phenotypes. These are quantitative traits that are measured usually in a numeric scale. You get a histogram, kind of height as an example. Now, this is how you shuffle up or restructure a population when you make them. Some specific variants in each parent could be brought together in a hybrid, and these colored chromosomes then are introgressed in the next generation, and a lot of both genetic and phenotypic variation is released. This is the traditional approach for QTL mapping. What you get is these populations of chromosomes that are recombinogenic, and we want to see which phenotypes may correlate with the color at any one locus. And we'll do a SNP scan here in a minute, possibly on a subset of phenotypes. This is the high-resolution data that we deal with now. This is Arabidopsis thaliana, and 1,000 genomes data set has been released in the last year or so. So measuring this set of lines in your favorite conditions is the rate limiting step. You can get the data is all up online. You can upload the data and put it right into a genome-wide association browser. So it's very exciting. Now, you see many phenotypes in that picture. How do you extract those quantitative measures? That's our current challenge. Now, SNPs are not living, in a free world. They all are inherited together along a chromosomal block, and this is what you get when you correlate not samples with samples, but the same data just transposed. And if you correlate SNPs with SNPs, see these white lines are rare variants or missing data that you can't correlate with anything else. But if you look, most SNPs are essentially inherited independently. That's the red section, but blocks of SNPs are generally correlated with each other due to linkage disequilibrium. And you really cannot separate which of those SNPs in a block is controlling the trait. The whole block will be associated. So you need better restructured populations that mix up and break down those blocks. So here's a real-world example of a flowering time gene in Arabidopsis that's been under selection in Sweden, and there's less haplotype diversity up there, panel B. But if you just look down there in panel D, you can see that there's a large block of linkage disequilibrium. So those SNPs then are inherited together, and it's related to this recently derived translocation. So it's complicated in the real world. Now I'm going to just pause right here and come back to another talk about tapping into genetic diversity after the next break. Thanks very much.